Can you hear me well?
Yes, absolutely, yeah.
Good. So I hope you still like me after this talk, because I'm not going to talk about what I promised to talk about. So I'm not going to talk about metabolic labeling. And the simple reason for that is that it's already published. And I really enjoy talking about unpublished work. So the thing I will talk about today is dynamics, though, so it's not completely unrelated. It's definitely about temporal analysis in single cells, but not metabolic labeling. So let me take a step back and go to where the field single cell sequencing came from, right? So if you think about 10 years ago, it wasn't possible, and there were basically two different fields in biology. There were what they call the omics people, and there were the single cell people, and I was, and I'm still one of those single cell people, basically sitting behind a microscope, making beautiful movies of cells, well, dividing, looking at a few genes at a time using GFP or RFP. And we were always very jealous of the omics people that were sequencing genomes and looking at thousands of genes at the same time. But of course, they could not look at single cells. So now we're in this very exciting area where we can do both. We have the best of both worlds almost. And we can do many genes and many cells. So that's really, really interesting. But of course, when something like this happens, there's always something that is sacrificed, and it's often the temporal information that is sacrificed. Before, when you were sitting behind the microscope, every second you could take a picture. Nowadays, when you do a single cell sequencing experiment, it's a destructive experiment. So you cannot really do, as mentioned before, many experiments on the same cell. It's just one experiment. So how do you figure out what happened in time? That's a big challenge, and which is, of course, the topic of this workshop. How can you retain information on temporal events when you're doing single cell sequencing? Now, we heard already beautiful talks on RNA velocity and metabolic RNA labeling. So these are two methods that allow you to basically, even though you do a destructive experiment, to look back in time what these cells were doing a few cell cycles ago. The thing I want to talk about is the third option here, which is how can you tell something that the cell many cell cycles ago? What was its lineage? How did that cell relate in terms of the lineage tree to the other cells? What is a sister of another cell or a mother? How do you figure out how the family relations were laid down during the life of that cell? So of course, this is a different topic, but it's also very much a question about dynamics. So what I'm going to do today is to talk about a system where, well, let me take a step back. So there's a very beautiful literature now on lineage tracing using single cell sequencing using CRISPR-based technologies, where the CRISPR can basically mutate a target into the genome. And in this way, you can really speed up the mutation rate. This, of course, works really well in model organisms and in cell lines, but for human tissue, this will be difficult. So we're really looking for new ways to also do this without CRISPR. So look at other marks in a genome that are heritable and informative about the lineage. There are basically two things that we focus on. I will show you data on this today. So one is copy number variation. Now, this is mostly relevant in the context of cancer, where a cell will sometimes lose a chromosome or gain a chromosome. And this, of course, is a heritable change that will propagate to the future. Somatic mutations is another way to do it. So these are mutations that happen, for example, during the S phase of the cell when the genome is replicated. A handful of mutations are introduced, and these, of course, are also heritable. It is a very difficult measurement because, as I said, there's a handful of mutations, and there's 10 to the 9th non-mutation. So it's really a needle in a haystack experiment, but these things are possible nowadays. So I want to focus on those two things, and the experimental system that I want to work with is kind of inspired by colon cancer. So how colon cancer works, if you read the textbook, is that it starts all with a stem cell that is still healthy and normal in the colon, and then accumulates over time a couple of mutations, which are called the driver mutations, that basically turn the cell into an uncontrollable dividing clone that starts the tumor. And then in the tumor, there is all kind of other events that happen that make the tumor even more heterogeneous. So some cells in the tumor will get another mutation or will lose another chromosome. And after some time, you have a very heterogeneous tumor. And some of these clones will then escape and metastasize through the body. So in an ideal experiment, you would like to sample this whole process at regular intervals, from the early point of the initiation of the tumor until the end of the tumor, or the end of the life of the tumor. Of course, in a real setting, in a clinical setting, this is very difficult. So what we do is we do the next best thing, and we use an in vitro system based on organoids. So organoids are basically cells that form a structure that resembles an organ, but can be grown outside the body, outside the organism. And what we did, actually, the group of Hans Klevers in our institute, what they did is they took healthy human colon cells and stem cells and grew them into these organoids. And then with CRISPR technology, introduced those, or the most famous form, driver mutations into those organoids. So now these organoids turned into invasive cells that basically can start a tumor in a mouse. So we start with these cells, and in addition to this technology, we also label these cells individually with a barcode. So we use a lentiviral system to label each cell with a barcode. It's basically a 10 nucleotide long code that uniquely labels every single cell in the organoid. Then what we do is we split this culture in 3 replicas, and we do a very long-term experiment, so the order of 25 weeks. And regularly then we take samples during the evolution of these organoids. And some of the time points we only bulk sequence the genome, but many time points we use single cell genome sequencing during this whole time point, during the 25 weeks of these experiments for 3 independent organoids. Actually, when I say we, I really have to say Leonard, who did basically all the experiments. So Leonard was a graduate student in my lab. Now he's moved on to the next step, but he really spearheaded the experimental part. And Buijs is a computational biologist in my lab currently, also a graduate student who did all the analysis. And the analysis is very challenging, as you will see, particularly for detecting these somatic mutations. This was all done in collaboration with Jeroen de Ridder, who was in the hospital in Utrecht. Okay, so what we're going to do, the experimental goal of this experiment is that when we have these organoid cells, that at many of these time points, we can measure 3 things in the same cell. The copy number changes, the somatic mutations, and then also to read out the lentiviral code of that particular cell. Of course, this lentiviral introduction is only done at one time. That's at the beginning of the experiment, right? We don't introduce these viral barcodes later. It's then at the beginning of the experiment, and then we wait 25 weeks. Of course, the somatic mutations and the copy number mutation, the changes, they happen during the experiment. Okay, let me first focus on the copy number changes. So I will not go through the details how we measure it in detail, but basically what we do is we take the genome of a single cell, we digest it with a restriction enzyme, and we ligate adapters to it that have a T7 promoter. And this T7 promoter will produce RNA when you do an in vitro transcription step. So that's basically, this last part is actually the same chemistry that is used now also in the 10x system, but this is now done for DNA. So what you see here are the different chromosomes in the cells, and these are individual cells. So this is cell 1 here, and when you see gray, it's basically diploid. So it means there are 2 copies of the chromosome, so it's a perfectly diploid cell. Now you can see there's some deviation. So this cell has also a loss of a piece of chromosome 1 and a gain of chromosome 9 here. And then another cell also has the same loss of chromosome 1 little piece, but has a gain of chromosome 8 and also a loss of chromosome 8. So you can make a catalog of many, many cells, how the copy numbers change. And you can already see, if you kind of compress all these cells on top of each other, if you look at thousands of cells, you already start to see copy number clones. So this is all the cells of all experiments combined. This is not one particular time point, but this is all the 3 replicas at different time points combined. And you see there is very clear reproducible copy losses. For example, chromosome 4 is lost quite a bit. And chromosome 9 is gained quite often. So this is interesting. So these data are quite sharp. If you look at one of these transitions in the chromosome 1, you can, if you're lucky, within 50 kb or so, determine these breakpoints. It also depends on how deep you sequence, but we can quite nicely identify the breakpoints and count the copy numbers. Now, these copy numbers changes in the copy numbers are not random. What's very interesting is if you take those organoids and you look at the losses and gains of the chromosomes, they're actually very similar to losses and gains that you see in real tumors from the colon. Right? Chromosome 4 is lost very often, and chromosome 13 is gained a lot. So there's a lot of correspondence between the chromosomes that are lost and gained in our organoid system and in real tumors. So that it seems to reflect the real physiological situation. Okay, so let me go to the somatic mutations. So somatic mutations, as I already said, that's a really tough one, and this project is going on for some time, and this is really causing us a lot of delay to really do this very carefully, because you can imagine you can interpret mutations, but they're not mutations. So there's a lot of things that look like mutations, but they're not mutations. Sequencing errors, mapping errors, all kind of other artificial things that are looking like mutations, but they're not. So one thing that really helps here is the idea that any somatic mutations that you're looking for, that you phase it with a SNP. So here in this slide, I'm using this notation of a GSNV. So this is a genomic SNV. So this is really a SNP. And this would be a somatic single nucleotide variant. So this is the somatic mutation I'm talking about. So what we do is we scan around SNPs for somatic mutations. And this allows you to really have an additional quality check. And we didn't invent this. We basically use these two methods called Bayes and Lira, basically the same idea, to phase somatic mutations with SNPs. And if you do this, you get much better quality data. And what's also very nice is that you can also identify if the mutation is not there. So for example, in this matrix, what you see, so there's single cells here in the columns, about 300, and the rows are different somatic variants, that we can actually call if the variant is there or not. So in red, it would be actually mutated. In blue, it would be the non-mutated allele. So you can really nicely measure both the absence and the presence of a somatic mutation. The last thing we measure in the same cell are these lentiviral barcodes. They're also in the genome. Remember, this is introduced at the start of the experiment only. And then we get this kind of plot. So what you see here on the x-axis is time. So this is actually measured in numbers of generations. So as I mentioned, this is 25 weeks of experiments. We go up to 100 generations, cell generations. When we start the experiment, you see there's many viral barcodes, which is of course what you want, because every single cell has been labeled almost with a unique code. And then over time, what you see is you can see selection. So after about 100 generations, there's only about 5 to 10 clones left, whereas we started with 50,000 or more. So you can already see in this culture that there is a lot of selection going on, and that there's certain clones that are more fit than others. And after 100 generations, a handful of these clones are left. So this is also a general lesson, I think, for people working with organoids, especially when there are cancer mutations in those organoids. There's a lot of selection in these organoids. So organoids, I think, at least the ones with the driver mutations, they're almost never clonal. I think every cell will be almost different in that organoid, which is interesting, but you have to realize that it's very difficult to make them clonal. Okay, so we have these three measurements now done in the same cell, and now we have this challenge, which is a major challenge, to really integrate them, these three data sets, into one data set. So I'll walk you through how we do this, and I'm pretty sure there are more elegant ways to do this, so please let me know if you have good ideas, because this is not only computationally challenging, but also how to present the data is actually very challenging, because we have three measurements in each cell. We have also time, we have replicas, we have all kind of different things happening at the same time. So I'm struggling also at the moment with how to present it to you, but this is the best way at the moment I think we can do this. So first we just start with the copy number information. So we ignore everything else, just look at the copy number information. Here on the x-axis is time again, in weeks. So 25 weeks is the end of the experiment. 0 is when we started the experiment. So this is where the lentiviral barcodes were introduced. And then I'm not showing all the raw data, but I'm just showing the reconstructed copy number tree from this. Each kind of band that you see here has a certain width. That width is basically how many cells are in that copy number state. So for example, at week 10, you can see that most of the cells are here in this part of the tree, which is the loss of chromosome 18. So after 10 weeks, there's already in this particular replica, most cells have lost chromosome 18. So again, the width of the band is proportional to the relative frequency of that particular copy number state. And so each of these branches corresponds to a certain copy number state. For example, here, this one branches off from the 18 branch, and also loses chromosome 4. So all the cells in this branch have lost 18 and 4. There's also a couple of these branches that we don't see anymore later, so that we interpret that these branches basically die out. So these cells have been outcompeted by other copy number genotypes. So this is, again, only the copy number information. Now I'm going to basically overlay the lentiviral information on top of it. So I do this with the color. So for each of these time points, of course, we have cells where we also have information about the lentiviral barcode. And that I'm indicating with the color. So different color means a different lentiviral barcode. So there was a different ancestor line that was created at the beginning of the experiment. And you can see that some of these colors, they die out. For example, at the end of the experiment, I only at this point see 2 different colors. And earlier on, there are more. So what's quite interesting and really confused us at the beginning a lot is that when you, for example, look at this clone here, so there's a major clone here. Now, if you think that that clone comes from one ancestor cell that at a certain point lost chromosome 18 and basically populated the whole, the whole tube with cells, that's probably not what happened, because what we see is we see at least 5 or 6 different colors here, which means that there's 5 or 6 different cells at the beginning of the experiment that during this evolution experiment lost chromosome 18 independently, perhaps. So that will be one interpretation, or it could be that already at time equals 0, already at the beginning of the experiment, these cells were already there. There was already a subpopulation that lost chromosome 18, and we labeled the members of that clone with different lentiviral barcodes. Right? So that is hypothesis 1, that the heterogeneity was already there at the beginning of the experiment, and we labeled those cells independently. If you look at the bulk copy number profile of the founder population, however, it looks pretty diploid. There are a few little glitches here and there with a loss of a little piece of chromosome 1 and 9, but most of the chromosomes are at a copy number of 2. So it could be, we can't rule it out, to be 100% sure, but it's unlikely that there were a lot of cells already there that lost chromosome 18. So that's possible, but we can't rule it out. The more interesting hypothesis is that the copy number changes are recurring during the evolution experiment. So rather than having one founder cell that creates a clone of chromosome 18 loss, there are many founder cells that basically lose chromosome 18. And this hypothesis is supported when we look at the somatic mutations. But before going there, I want to show you one more thing. Is when you go to the end of the experiment here, you can see here that I have chromosome 18 and 4A and 4B. What I mean by 4A and 4B are the 2 copies of the paternal and the maternal chromosome 4. So we have also allelic information, since we map the SNPs and we can also haplotype them. We know which allele it is. And what you can also see here is that sometimes you lose sometimes the cells lose chromosome 4A, say from the father, and sometimes from the mother. So already from here you can see that at least those 2 events are independent. But then also here you can find 2 lentiviral barcodes at the end, which means that these 2 founders lost both chromosome 4 independently. Or very, very small probability that both 18 and 4 were already lost at the beginning of the experiment, but we didn't see that in the data, so that's unlikely. So we think what's happening is during these experiments, these chromosomes are lost recurrently quite often. And this is also confirmed by the somatic mutation. So what I'm showing you now is a similar plot. So on top of the copy number 3, I'm plotting if a certain mutation at a certain location in the genome, so this is really a single nucleotide, is there or not. If it's red, it's mutated. If it's green, it's still the reference value, so it's the unmutated version. And what you can see is that, for example, in this clone, the clone that has both lost a copy of 18 and 4, you can see both cells that have the mutation and cells that don't have the mutation. And actually you also see it here. So this also means, and another thing which is interesting is that this mutation is not observed before. So most likely that mutation is introduced during the experiment, created a founder that now lost chromosome 4, but also the cells that don't have the mutation lost chromosome 4. So it happened multiple times. It cannot be the same cell that lost it and then generated another clone. So this shows, and this is one particular nucleotide, but you can look at many other nucleotides that basically resemble the same behavior. So these are three different positions that basically show the same behavior. Okay, so let me wrap up. So this picture I showed you before, the textbook picture of generating this tumor heterogeneity, where all these shades of color indicate different genotypes in this tumor. What we think is happening, so is probably true, but there's also quite a bit of similarity between a lot of these clones in terms of the copy number, at least. So of course we're not 100% sure if this copy number drives the aggression of this cancer, but what is clear, or our hypothesis at least, is that it seems that many of these clones arise independently and have exactly the same copy number states. Now, if you go to clinical data, it also actually is visible. So this is a histogram, basically, two-dimensional. I'm almost done. Yeah, so this is a histogram where patient data is shown for colon tumors that lost both chromosome 4 and 18. So you can see that's where these patients are. They both have lost 18 and 4. But you see that in this part of the face space, there is not that much tumors found. So this suggests that tumors that just lose chromosome 4, they don't exist that often. They first have to lose chromosome 18, and then they would lose chromosome 4. So this is basically also what we see in our data. And these patients, unfortunately, have a worse survival probability, the ones that lost chromosome 4 on top of chromosome 18. So this suggests that these are not really, these tumors are quite aggressive. So let me conclude. We developed a single cell DNA sequencing technology that can read out the copy numbers, the somatic mutations, and also this viral barcode that we introduced at the beginning of the experiment. We see there is quite a bit of selection during this culturing. So that we start with a very heterogeneously labeled population that ends up with only 5 or 6 clones after 25 weeks. You know, with this is important for organoid people that think about clonality within these systems. I think they're almost never clonal. If we look at the different measurements, we can reconstruct the order of oncogenic events. And we see, for example, a loss of 18 before 4. And what we also see is that it's very unlikely that these chromosome losses all happened in one founder cell. So our hypothesis is that this is a very recurrent phenomenon, and there's many cells that independently lost these chromosomes and show a very similar karyotype, but that it's not coming from one founder cell, but from many founder cells. And many of the aspects that we measured, they are also reflected in clinical observations. So this is all unpublished. So if you have any good feedback for me, let me know. If any of the editors in the audience are excited about it, send me an email. And of course, I want to thank the lab because they really make this happen. Very enthusiastic group of people that are really hardworking and very smart. Very grateful to be working with these people. And also want to thank the funding sources. Thank you very much.
Thanks, Alex. You have a lot of questions. Sorry for interrupting you. Sebastian, you want to start? Take the microphone.
Hi, I had a bit of a question regarding the several founder cell situation. Could it be that, like in vivo, when you have an actual cancer case and it's like fighting against the immune cells, that cancers which would have several different lineages would be like more fit because they would have more variation, therefore more likely to survive, and therefore like perhaps that would like play a factor in like whether a cancer becomes an actual cancer or whether if it's just one lineage that a cancer would perhaps die out or something?
Yeah, it's a very interesting question. To be honest, I wouldn't know. So of course, this system is very simple in that sense, is that it's all epithelial cells, right? There's only one particular tissue. What happens if you have multiple cell types and multiple tissues in there, like immune cells, is hard to extrapolate, from the data that we have. I think that one thing we can learn from this, I think, is that the fact that these copy number changes are so recurrent, also I think points to if you want to try to avoid it, you can really focus on those particular copy number variants. So if you can find vulnerabilities that would correspond to these particular chromosomes, it would be perhaps a good therapy to go in that direction, because you know it will happen many times. It's not only that once you have to stop it, you have to do it multiple times. But how it exactly works in with multiple tissues, I think experiments have to be done. And the organoids are actually quite promising to do co-culturing with immune cells, for example.
Thank you very much.
Yeah, Casper, your turn.
Yeah, so in your conclusions about sort of certain lineages dying out in your organoids, are you worried about sort of they grow in some spatial matrix, right? So you're not are you worried that you're just sampling kind of, you know, part of the cells and that they're sort of in there in some other spatial location that sort of moves over time? I don't know much about organoids, and I don't know much about how you sample from them, but
it's a good point. I think the sampling, of course, we can't sequence every single cell. That's the problem, right? So there's always the small populations will suffer from sampling statistics, and some of these dying off populations might just be that they're still there, but they're not visible because we didn't sample enough. Of course, you know, these organoids are refreshed all the time, right? So they grow to a certain size. Then, in a sense, we basically take 1/4 for one experiment. So we divide that culture into 4, and 1/4 is continued, is basically go back in culture, and the other 3 parts are for different experiments. So, but you could also lose at that point when you split the population. You could also lose a population. So I think it's not a problem for the abundant populations, but for really detecting small populations, you might lose them like this. But there's really no other way to do it.
Scott, do you want to take the microphone?
Yes. Hello. Thank you for the talk. I just want to know, ask like why chromatin 18 is more prone to loss in this like more recurring situation, and what will be your next step to like T cell? Is there something similarity in the founder cells, or like what do you like speculate that will happen?
Yeah, no, that's a very important question. So I don't know the answer. We tried. So actually we have several clones that don't lose the full chromosome 18, but they lose a little piece of it. And so that chromosome 18 is very big. You know, if there's many genes on that, so it's very difficult to form a hypothesis just on the whole chromosome. But we have been looking at some of the genes that are on a smaller part and are still having this selective advantage. It's still quite a large part. We didn't pin it down yet. I'm not sure if we ever will. But if I would start there. I would start to make clones that survive and lose smaller and smaller pieces of chromosome 18 until it's manageable to start to, for example, knock down or knock out or overexpress certain genes that are still left on this piece of chromosome. But at the moment, we have no mechanistic understanding of this. It's interesting that it also happens in real tumors. So 18 is a very frequently lost chromosome in colon tumors. So most likely there's a very important gene on there that, or many genes on there that explain this, but we don't know yet.
Yeah, you still have 4 questions. Let's make them short, like short question, short answer. Shalef, you want to take the microphone?
Yeah, can you hear me?
Yeah, we can hear you.
Amazing talk, Alexander. I'm wondering, are there any known tumor suppressors on chromosome 4 and 18? And also, there is some discordance between the copy number variation and the tissue and the organoids. I'm wondering if the chromosomes that differ between the organoids and the tissue harbor some genes related to the tumor microenvironment or interaction with the immune system, which is absent in the organoid.
Yep. Very good to see you, Shalef. Yes, the only thing I can say on the SMET is on SMET 4 is on chromosome 18. So that will be my first, and you remember that's one of the ones that's mutated, right? So that will be my first guess, but it's very similar to the previous question. Again, it is, these chromosomes are huge, right? So there's so many genes on there, but this would require much more work, and I think looking for deletions that are not full chromosome, but parts of the chromosome, this will help you. And we didn't really make any effort. We didn't make any progress there.
Thanks.
So in the order, Natalie? Natalie, are you here?
Yeah, hello, can you hear me?
Yes.
Yes, my question was related to the organoids that you used, and I was wondering if you used iPSCs that were derived from patients with colon cancer, or if they're just...
No, so these are biopsies that were taken from healthy patients. And so this is not from stem cells, right? So these are the adult colon stem cells that the organoids were grown from. Yeah.
Okay.
Yeah. So so next one, Florian, you want to take the microphone? Florian.
Yeah.
Florian, yeah.
Thanks. Yeah, it's a short question. Basically, whether you're also exploiting mitochondrial SNVs or to what extent you're exploiting them.
Yeah, very interesting direction. Now we haven't been looking at them.
Great. Jon, welcome. Take the microphone.
Yes, now it should work. My question obviously is whether you consider genomic and epigenomic crosstalks in your system, because of this frequent loss of chromosome 18, that might have a reason also based on epigenomic changes that lead to a more frequent loss of chromosome 18. And the second question, which I didn't pose here, is you were looking into a loss of chromosome or a gain of chromosome 19, I think, in the beginning, but later you didn't mention it anymore. Is that?
Yeah, so the epigenetic part, again, also we didn't really explore this. I think that's all great suggestions for future work. Let me see with 19. The one I think might be 9, right? Oh.
Was it 9 or 19? It was 9. Sorry.
Yeah. So this is one which is actually in the founder population. So that's what.
Okay. Good.
Yeah. So you can see it here. You see? So this.
I missed it. Sorry.
No problem, no problem. But so this was probably already there at the beginning of the experiment.
And a cool story, really good.
Thanks so much, Alex, that you have given like another perspective, another time scale, and that was really important for us today. So thanks for bringing that along the way.